Hello everyone. It is my honor to introduce our article published in iMeta, entitled Environmental Chemicals and the Tumor Immune Microenvironment. This work was accomplished through collaborations among Hainan University, Huazhong University of Science and Technology, Chongqing Medical University, the University of Oslo, and the University of Galway. This article systematically summarizes the regulatory effects of common environmental chemicals on the tumor immune microenvironment, TIME, explores the underlying complex mechanistic networks, and proposes current research limitations and future directions for this field. We provide a new scientific reference for deepening the understanding of the immunological mechanisms underlying environmental carcinogenesis and for exploring effective strategies applicable to cancer prevention and treatment. The tumor immune microenvironment TIME is shaped by a complex interplay of epigenetic and environmental factors, and plays a central role in the initiation and progression of human cancers. While intrinsic regulators of the TIME have been extensively studied, the contribution of extrinsic factors, particularly environmental chemicals, has received far less attention. Populations are routinely exposed to a wide variety of environmental chemicals. After entering the human body, these substances accumulate in several vital organs and ultimately disrupt normal physiological homeostasis. Within tumor tissues, the accumulation of environmental chemicals can remodel the TIME and interfere with antitumor immunity. Therefore, we summarize the impacts of common environmental chemicals on the TIME and the underlying molecular mechanisms. First, environmental chemicals generally function as TIME modulators. All of them can remodel the TIME through multiple pathways. The overarching trend is to alter immune metabolism within tumor tissues, promote the infiltration of immunosuppressive cells, and impair the function of cytotoxic immune cells, thereby inducing an immunosuppressive state and facilitating tumor progression. Second, the effects of environmental chemicals on TIME remodeling exhibit high complexity and bidirectionality. For instance, inorganic arsenic typically promotes an immunosuppressive microenvironment, whereas arsenic trioxide exerts antitumor effects by inducing oxidative stress and immunogenic cell death. Third, environmental chemicals act through multiple intertwined molecular mechanisms. These mechanisms interact with one another and collectively form a complex regulatory network. Fourth, exposure to environmental chemicals may affect the efficacy of clinical cancer immunotherapy. Environmental chemicals-induced T-cell exhaustion and upregulation of immune checkpoint molecules may compromise the intact immune response and cytotoxic functions required for tumor immunotherapy, thereby negatively impacting therapeutic efficacy. This research field currently still has numerous limitations. First, many findings are derived from in vitro cell experiments, while human epidemiological data and clinical validation that better reflect real-world conditions remain relatively scarce. Second, insufficient attention has been paid to the initiating molecular events after environmental chemicals enter organisms. Third, the application of high-resolution analytical techniques such as single-cell and spatial transcriptomics in environmental toxicology remains limited. Fourth, environmental chemicals typically exist as complex mixtures in real-world scenarios, yet most current studies focus on single substances or simple combinations. Based on the above challenges, we propose the following priority directions for future research. 1. Use models closer to the real time, such as patient-derived materials and humanized mice. 2. Employ biochemical methods to map molecular initiating events, comprehensively identifying the target sites involved in the early stage of environmental chemical action. 3. Integrate single-cell multi-omics data with spatial transcriptomics to precisely locate susceptible immune subpopulations. 4. Construct representative mixture models and apply exosomics analytical techniques to deeply dissect the synergistic or antagonistic immunomodulatory effects produced by complex chemical mixtures, thereby addressing real-world complex exposure scenarios. In summary, this article breaks through the limitation of previous studies that mostly focused on intrinsic regulatory factors, concentrating instead on the critical remodeling role of exogenous environmental chemicals in the TIME, thus providing an important new perspective for research on the regulation of tumor immunity. It systematically summarizes the common effects and complex mechanisms of action of environmental chemicals as TIME modulators, offering a new theoretical framework for understanding TIME alterations induced by environmental exposure. Finally, grounded in the complex characteristics of real-world environmental exposure and based on a review of current research limitations, this article proposes clear, systematic, and forward-looking future research directions.